Students. Good afternoon, students. Today we are going to observe human cheek cell under a compound microscope. Apparatus needed: a clean toothpick, methylene blue solution, glass slide, water, needle, compound microscope and cover slip. And after this experiment, what you will learn? That animal cell is different from plant cell. So students, let us begin with the experiment. Now in order to observe human cheek cell under a compound microscope, we need watch glass, glass slide, toothpick, forceps, methylene blue solution, cover slip. And lastly, we are going to use compound microscope. Now with the help of marker, we are going to mark it on a glass slide. And we are going to turn it. Now at the center of glass slide, we are going to keep fluid which we have collected from chicks. And as you know, that chick is cells are transparent. So we are going to add few drops of methylene blue solution so that we can able to study cell. We can observe it. So we have added few drops of methylene blue solution. Now with the help of cover slip, we are going to keep it on a glass slide in such a way that there should not be any air bubbles inside it. And we are going to keep this glass slide on the stage of compound microscope. And now we are going to study it through compound microscope. So what you have observed? You will see that nucleus is present in each and every cell. And cells are irregular shape. And more what you will see, that human cheek cell is not having a distinct wall, that is cell wall. So nucleus is present in each and every cell. So when you have observed human cheek cell under a compound microscope, so it will look like this. Nucleus is having a thin membrane, and that we are saying nuclear membrane. And more what you have observed, that in each and every cell there is a spherical structure, and this spherical structure we are saying nucleus. So nucleus is present in each and every cell, and it is having a thick fluid, and that thick fluid we are saying cytoplasm. More what you have observed, that it is having a thin membrane, and that thin membrane we are saying cell membrane. So students, you have observed onion peel cell and human cheek cell under a compound microscope. Onion peel cell plant cell and human cheek cell animal cell. Now we are going to learn the difference between plant cell and animal cell. So in plant cell, you have observed that it is having a rectangular shape and it is lying side by side to each other. Whereas human cheek cell, cells are irregular shape. And second difference what you have seen, in plant cell and human cheek cell, that plant cell is having an empty space at its center, and that empty space we are saying vacuoles. Whereas in human cheek cell, what you have observed at its center, that nucleus is present at the center of each and every cell. And what third difference you have seen? That plant cell is having a distinct wall, and this distinct wall we are saying cell wall. Whereas in animal cell, you will see that cells are having a thin membrane, and that thin membrane we are saying cell membrane. So students, what three difference you have learned? In plant cell and animal cell, that plant cell is having a rectangular shape, whereas animal cell is irregular shape. Plant cell is having vacuoles at its center, and that we are saying vacuoles, and human chick cell is having nucleus at its center. Third difference what you have learned, that an plant cell is having a cell wall, whereas human chick cell is having a thin membrane, and that thin membrane we are saying cell membrane. So after this experiment what you have learned, that animal cell is different from plant cell. Thank you.